I am pleased to introduce our speaker, Dr. Jerry Parks. He is the group leader for molecular biophysics in the Biosciences Division at Oak Ridge National Laboratory. He's also a senior R&D staff scientist. He uses his expertise in computational chemistry and bioinformatics to determine what certain proteins and enzymes look like, what they do, and how they work. He will speak today on structure-based antiviral drug discovery, and he will talk about the COVID virus. A native of Texas, Dr. Parks holds 3 degrees in chemistry. His BS degree is from Texas Christian University, his MS degree from Southern Methodist University, and his PhD from Duke University. In both 2013 and 2020, Dr. Parks received an ORNL Director's Team Award and a UT Patel Award. In 2013, he and his team were cited by the ORNL Award for groundbreaking research in biotic and abiotic mechanisms of mercury methylation in the environment, leading to high impact publications in Science and Nature Geoscience, 2 prestigious scientific journals. The UT Patel Team Award cited him and others for the discovery of genes and proteins required for bacterial mercury methylation, the process that we know is going on in our local East Fork poplar trees. The ORNL Directors Award for 2020 cited him and the team for revolutionary advances in the area of microbial isolation techniques that promise to usher in a new era of microbiome studies, an area of current significant interest in both health and agriculture. Please join me in welcoming Jerry Parks.
We have to back up.
Thank you very much for the introduction. It's nice to be here again. I think this is my 3rd time to speak to you all, so I'm glad to do it. I was going to say feel free to interrupt me at any time, but that apparently has been forbidden. But if you have a question, if you would like some clarification, I won't get mad at you. You just have to deal with the organizers. Okay, so I've been at ORNL since 2008. I came as a postdoc. I was hired by Jeremy Smith. I believe he's probably given some talks here a couple of times as well. And shortly after that, I became a staff scientist and started working on the mercury research that you heard a little bit about. Some of you are very familiar with that, maybe even more familiar than I am. And over the years, my interests and expertise have morphed a little bit into different areas. I learned about bioinformatics, which is studying protein or gene sequences and trying to figure out different aspects of how biological systems work. I'm really a quantum chemist by training, so that means I use quantum mechanical approaches to model molecules and their reactions, but I haven't done so much of that over the last few years. I've really gotten involved in protein structure prediction, and we may talk about that if we have time toward the end. But there are some approaches that have come on the scene in the last couple of years that are just amazing. So like I said, we may talk about that if we have time. So today I'm going to talk about structure-based drug discovery. And I will define what that is, hopefully adequately, but if not, wave at me and I'll go into more detail. Okay, so the coronavirus, everyone's familiar with it. Probably we've had it. I have a couple of times. But I'm doing well. That's good. I'm thankful for that. So let's talk about the SARS-CoV-2 virus. So this is a picture of it. Let's see, do I have an arrow? Yes. What I want to point out is, I think everyone is familiar with the spike protein. We've all heard about the spike. But what about the rest of it? What about the guts inside of it? So this gray stuff is nucleocapsid protein that surrounds the single strand RNA. So that's the RNA genome of the coronavirus. And some other things that I want to highlight, maybe it's a little difficult to see, but there is something called NSP3. We're going to talk a lot about that one. There's the main protease. We'll talk a lot about that one. And then NSP12 or the RNA dependent RNA polymerase. Those are some ones that I'm going to highlight. So RNA is ribonucleic acid. It's converted, the sequence of RNA is converted to protein sequence. And those proteins fold up into three-dimensional structures and have some function, okay? So let's talk about the coronavirus RNA genome. So this is just a very simplified diagram of what's in that genome. So it's about 30,000 bases, 30,000 RNA bases. So that's A, U, C, and G. And that gets converted into protein. And so you can see that these are the genes. This thing is called an open reading frame. That just means it's the part of the RNA that gets read and converted into a protein sequence. And then some other things down here that are not important for the current presentation. But these open reading frames, they encode proteins in SP1, 2, 3, 4, and so on, all the way to down here at 16. Well, what are those? NSP stands for non-structural protein. That means that it's not just like a scaffold. It's not the outer envelope of the virus. It's functional. It's an enzyme that's doing some reaction. And so there are several different ones. But then there are the structural proteins that are down here, and the spike protein is the most notorious of those. So there's another open reading frame, and the main thing I want to talk about there is NSP12, which is also called RBRP, or the RNA-dependent RNA polymerase. Okay? Hopefully that introduces you just a little bit to some of the proteins that are encoded in this viral RNA genome. Now, let's talk about some drugs that are available on the market. Probably you've all heard of remdesivir. This is the structure of it. And another one that's a little less well known, but maybe you've heard of it, is molnupiravir. Both of those, they kind of look like RNA molecules. They have this sugar and then this base-like group here. And so you might expect that they target the RNA-dependent RNA polymerase I just mentioned. Now, this one, nirmatrelvir, who knows what that is? Has anyone taken it? Paxlovid?
Yeah.
Okay. All right, yes. So Paxlovid is nirmatrelvir and another compound that work together sort of synergistically. But this is the main drug. I haven't taken it, but my mom took it and it really made a big difference. So this is a big one. All right, and it targets the main protease. So this is another view. It's just a sort of a different view. This time, instead of being shown sort of schematically, these are experimentally determined structures. So since 2020 or so, scientists around the world started characterizing these proteins in terms of their three-dimensional structure, either with X-ray crystallography or cryo-electron microscopy. And so these are just some of the example structures that have now been published and are available. You could all download them for free if you wanted to after this meeting. I'm sure you won't do that, but you could. All right, so just going back to some proteins that I've already highlighted. Down here is the structure of the RNA-dependent RNA polymerase right here. And this yellow and orange, that's RNA bound to it. And so it's a polymerase, meaning that it replicates RNA. It replicates the genome. And it makes sense that you would want to target that to stop the virus from replicating. Next is the main protease that I just mentioned. This is where Paxlovid does its thing. Nirmatrelvir binds to that and inhibits it. And the next one is one that I'm particularly interested in. So this big thing, it's really all one piece, but they've sort of dissected it a little bit. This is NSP3, and the part that they've cut off is called the papain-like protease. So I mentioned the main protease, and then this is another protease. What do proteases do? They chop proteins, okay? So that you have a long strand of protein, they chop it in certain places, not just indiscriminately, but at a certain spot. And they recognize something about a specific protein and chop it at a certain site, okay? So let's go back to this diagram. Now, you see I've drawn these little triangles here. Those are the cleavage sites. So this initially, these large peptides or polypeptides are produced, but then they need to be chopped at certain places so the individual proteins can fold up into their functional forms. And so PLpro cleaves at 3 sites. And the main protease, which I sometimes just refer to MPro, so papain-like protease, main protease. The main protease cleaves these long strands at more sites. So it makes sense that Pfizer might want to target the main protease because it does more stuff, right? But why should we also think about targeting PLpro? All right, here's our diagram again. These are all the proteins that get chopped and produced by the main protease, shown in this blue thing I drew here. Main protease, just these 3. However, PLpro is just these 3. But PLpro also cleaves human proteins. So when it's inside of our body, it messes up our immune system. It cleaves these protein conjugates called ubiquitin, which are ubiquitous, as you might imagine, and something called ISG15, which is interferon stimulating gene 15. Interferon is involved in the immune response. So one of the ways that the coronavirus messes up our bodies is by PLpro messing up our immune response. And so taking these 2 things together, it makes a lot of sense that we might want to target PLpro with a small molecule drug. Okay, you could say, well, we've already got Paxlovid, why do we need another one? Well, what about when these proteins or the genome mutates and the proteins mutate, and maybe there's drug resistance? It's always good to have something in your back pocket so that you can have alternative treatments. So for example, antibiotics, right? We all know about antibiotics and how you have to be careful using them because bacteria mutate and these certain existing antibiotics lose efficacy because the bugs just find a way around them, okay? So that's another reason that we might want to come up with additional coronavirus drugs. So now I mentioned structure-based design. So now let's look at some structures. This is PLpro It's part of that larger thing I showed you called NSP3, non-structural protein 3. But we're just gonna focus on that part of it. And this, so that's the part that's shown in this sort of tan color. In cyan is ubiquitin. That's a human protein. So we've got a human protein bound to a coronavirus protein. And this is an X-ray crystal structure that someone determined about 3 or 4 years ago. And I mentioned that these protease enzymes cleave at a certain site. They chop up proteins at a certain site. And in this case, PLpro cuts ubiquitin at this LRGG sequence. When you have leucine, arginine, glycine, glycine, PLpro binds to that and chops right after that glycine. So it goes this way, it goes right to left, but the way we read it off would be LRGG. Okay, so we know how ubiquitin, this human protein, binds to PLpro. And then over here on the right, we have PLpro, but in this case, it's bound to a small molecule inhibitor. So what is an inhibitor? It's a molecule that prevents an enzyme from doing whatever it is that it does. And so we want to develop an inhibitor that prevents PLpro from chopping the viral proteins and from chopping human proteins. That's the whole point of drug discovery. This little molecule, it's called GRL0617. It was identified back shortly after the original SARS virus came on the scene. So that was, if you remember, I think about 2013 or so. And that didn't really make it to the United States, but it was in China. So a lot of people started studying it. And a group in Indiana discovered this molecule and realized that it inhibits the PLpro from the original SARS virus. Well, fortunately for us, it also inhibits the SARS-CoV-2 PLpro. And so here's a structure of it. And you can see that it binds, it's much smaller than ubiquitin, but it binds in pretty much the same spot. So that's good. What can we do with that? Well, let's zoom in a little bit. And what we see is, here's that molecule again, GRL0617. Here's a chemical diagram on the right. And what we notice is that, you see I've drawn this little dashed line in yellow, pointing to cysteine 111. Well, cysteine is an amino acid that has some unique properties. In this case, it is the group, or the functional group, or the moiety, whatever you'd like to call it, that does the cleavage or the chopping reaction. So this thing is reacting with whatever protein comes along and handling the chopping. So that's important because it's reactive. And it's about 7.5 angstroms away from the closest atom in our inhibitor. And so that's what gave us an idea. Could we take this inhibitor that's already pretty good? So down here on the bottom right, it has an IC50, inhibitory concentration of 50%. So that means the half maximal inhibitory concentration of 1 micromolar. So lower is better, micromolar is good, nanomolar is better, picomolar is a lot to ask, but there are drugs that are down in that range. But we'd like to lower that number from 1 micromolar to somewhere in the nanomolar range, okay? Keep that in mind. Can we do that by modifying this existing molecule? Now, just a bit of history. I told you this molecule already existed way back to 2008. That's when it was published. In 2020, we got started working on this project, and we wanted to develop something quickly. We didn't want to start from scratch. So we said, let's take what we know. This is the best molecule that exists at the time, and let's try to make it better. And that was our strategy, okay? So just going back to ubiquitin that I showed you a minute ago, this is a human protein, and that LRGG sequence of amino acids. Here's the leucine, arginine, and glycine and glycine. And then there's that cysteine, and you see that it's forming a bond with that last glycine. That's a covalent bond, okay? It's not just sitting there in space, it's actually formed a chemical bond with the enzyme. So we want to use that approach to try to make our molecule better. So again, there's just another view of that distance between the inhibitor and the cysteine. What we decided to do was to try to make something that's kind of like this GG, glycine, but not exactly. We made a linker that's going to link the original core of the inhibitor over here on the right to this cysteine. But then we also need a reactive group or an electrophile, so something that will be attacked by that cysteine and form a permanent bond. And when we do that, we totally inactivate the protein. So that's a pretty good strategy. And so we ended up designing quite a few molecules that were based on the original non-covalent inhibitor with this linker and this reactive group. And this is the chemistry. We have a nucleophilic attack that then forms this covalent adduct. And that was our strategy. So before we, well, I should mention, I'm a computational chemist. So I don't work in the lab, I do computer stuff. And how does that come into play? Well, we can predict how a molecule will interact with the protein using an approach called docking. So it's virtual screening or computational docking. These essentially mean the same thing. And so this is 50 different snapshots of the protein. And you see there's a lot of wiggling going on in this blue part and some over here in the green part. Not so much up here in the red and orange areas, but that's where the molecule binds. And so what we did was we took all these wiggly snapshots of the protein and docked the molecules that we designed in our brain and tested them on the computer. And we docked them first non-covalently before a reaction has occurred, because the molecule has to bind in the right place before the bond can form. So that's non-covalent docking. Then we made the physical bond between that cysteine that I talked about and the inhibitor, and made sure that that didn't mess things up, that it could still interact in a favorable way. And so we used one of the computers at ORNL. This was not actually the Summit supercomputer. This was a smaller computer cluster that we have. And we estimated how favorably our molecules would interact. And then we prioritized them for synthesis and testing. So I went to my colleague, Brian Sanders, and I said, hey, these look pretty good. Can you make them? And so he made a scheme to produce these molecules that we designed. And just here on the bottom right, you can see this molecule is bound in that site non-covalently. And if I advance the frame, changes color for some reason. But anyway, now we've got a bond here. So I think I colored it differently just to distinguish between non-covalent and covalent. And the molecule still binds in the same place, which is good. And it was scored to have a favorable interaction. All right, so let's look at some of our molecules. We then tested them in the lab. This was done by a former colleague, Stephanie Delaney. So just to remind you, this measurement of inhibition was 1 micromolar, and lower is better. So this compound, I'm going to talk a lot about compound 7. It's 0.094. So, and that's micromolar, or if you convert that to nanomolar, it's 94 nanomolar. So it's it's an order of magnitude better than this one, give or take. And then a couple of other molecules were this one's maybe not quite as good, compound 8. Compound 12 was very similar. These numbers are essentially the same, 0.9094 and 0.98. But this one, compound 12, turned out to be toxic. So we sort of discarded that one and continued with compound 7. We worked with a private company in Pennsylvania called Progenera. It's maybe cut off here in the top right, or, yeah. But they tested not just the chopped off part of NSP3 that we worked with. They tested the entire NSP3 protein. And this is important because let's say that our molecule inhibits just PLpro by itself, but it can't interact when it's in the context of this full complex. But what we found is it was an excellent inhibitor. And you see the numbers here, 0.049 micromolar. So as good or better than our tests at ORNL. And that's in comparison to the side-by-side testing of this original GRL 0617 molecule. This time it's 5 micromolar, 1 micromolar, 5 micromolar. It's essentially the same number because of the error bars and so on. But so we're definitely doing better, right? So it turns out that adding the linker and this electrophile or this reactive group improves the potency of our inhibitor. So then we worked with a group, well, we worked with 2 different groups, one at UT Health Science Center in Memphis and another lab at Utah State. And both of these have a BSL-3 facility. So that's biosafety level 3, which means that they can work with coronavirus and other awful viruses that we don't touch at ORNL. And so I just want to highlight a couple of things. Everyone knows that when coronavirus first showed up in the US, it was what we call sort of the original strain, but then, and that's sometimes referred to as USA Washington 1 2020. But of course, a bit later on, we experienced the Delta and then lots and lots of different Omicron strains. And I think that the ones that are going around these days are descendants of Omicron strains. So we sought to know, we wanted to know how good our compound works against the different strains. So these are virally infected primate cells. So basically a certain kind of monkey cell, not the whole monkey, just cells that were taken from a monkey. And he didn't mind, but anyway. So you can see, now this is, instead of an IC50, this is an EC50. And in the Washington strain, if you may be able to read that, I'll just tell you it's 0.068 micromolar. So that's great because this is not just the enzyme by itself in the lab. This is in the context of infected cells, and that means our molecule is getting into the cells and doing its thing. And so that's a great result. And then I didn't really highlight it, but this next column, CC50, this is a measure of cytotoxicity. What you want is for your molecule not to be cytotoxic, obviously. And so these numbers are greater than 10, greater than 100. They're sufficiently non-toxic that there's a therapeutic window where the molecule has efficacy and isn't toxic. That's what we're shooting for here. All right, but then we go to Delta strain, and now it's 0.29 micromolar. That's not bad, it's still good, but we don't know why the Delta strain seems to be less affected by our inhibitors. That's an unanswered question, I can't tell you why. And similarly for the Omicron strain, 0.68 micromolar, still good, but disappointing, I guess, in a sense. And it does not appear to be because PLpro is mutated. There's something else going on because I'm pretty sure that PLpro is essentially identical in these 3 strains. But that's all I can say. So we worked with a group at Stanford and the Stanford Linear Accelerator Center, which is adjacent to the Stanford campus. This is Soichi Wakatsuki, and they took our compound 7, a favorite one so far, and they incubated it with PLpro, and they were able to solve a crystal structure of our molecule bound to the enzyme. which is really nice because it shows that we knew what we were doing. We designed a molecule and it actually did what we thought it would do. And so here, this is maybe a bit messy, but what I want to point out is this is a superposition of our docked model, the virtual screening that we predicted is how the molecule should bind, and that's shown in gray, and then the X-ray result. And you see that they superimposed very nicely. Now, it's sort of cheating because we already knew how this part of the molecule should bind, and we also knew that there would be a covalent bond over here. So we couldn't really stray too far, but anyway, it did what we hoped, that the molecule would do. So something else that's really important in drug discovery is off-target effects. So this is related to toxicity. Let's say you have a molecule that's great in the lab, then you give it to a human or a mouse or whatever, and it inhibits other proteins that you don't want to be inhibited. Well, that's bad news because then your drug needs to go back to the drawing board. And so what we did was we tested it. We paid a company to test some of our molecules. We'll just focus on the last column here, compound 7, to test it against these 7 proteins. And why did we choose these 7? Because they're very similar to PLpro. So PLpro has some proteins that look a lot like human proteins. That could be a bad thing. But it turns out that none of our molecule, none of the 3 that we tested, in particular compound 7, none of them was an inhibitor of any of these proteins. So that's great. Now, we didn't test all 20,000 proteins encoded in the human genome, but this is a start because these are ones that are likely to be the most similar and very well could be effective. So yes, higher numbers are better. So again, going back to this structure-based approach, we tried to rationalize those results. Well, we have crystal structures of some of these proteins. And so we can take our molecule docked to or bound to PLpro and superimpose it on these related human proteins and see, do they overlap? Is there stuff that gets in the way that prevents compound 7 from binding? And then it turns out that yes, this stuff shown in yellow here gets in the way of where our compound 7 would bind if it interacted with that human protein. And so that's why our compound doesn't inhibit one of these proteins. And then there's another example here. There are just severe clashes. So that our molecule, compound 7, just can't bind in the same way to the human protein that it binds to PLpro. So that's great. That's a design strategy that we took to limit off-target binding, which leads to toxicity. Okay. Another thing we have to think about is, does the molecule get broken down by our body? When you take a pill, it needs to get to the right place before it gets excreted or metabolized. Well, it turns out that this compound gets broken down pretty quickly. It's cleared by the human liver. And if you think about Paxlovid, I mentioned that it's 2 molecules. One is nirmatrelvir, and the other one is ritonavir. But what is ritonavir? It is a molecule that inhibits liver enzymes. And so you have to take them together so that nirmatrelvir doesn't get metabolized too quickly. Well, this one gets metabolized even more quickly than nirmatrelvir would. And so you can see this intrinsic clearance value is 138. Well, what does that mean? Well, it needs to be below 40 or 50. So we need to figure out a way to keep our molecule from getting broken down too quickly. And so we did an analysis. This is with our collaborator, Lori Ferrens at Northeastern University. This is just a sort of a quick calculation that predicts the sites of metabolism. So liver enzymes like to chew up aromatic compounds like this naphthyl group up here. This tertiary carbon is also very susceptible to degradation. This methylene group here, and then this ester over here. These are all sites that are readily broken down, but fortunately our electrophile and our linker, except for this part, seem to be pretty stable. So how would we go about improving our molecule? Well, we published a paper about a year ago, and we suggested some ways to do that. And one, this is that tertiary carbon. We can replace it with a cyclopropyl, this triangle-looking thing. This aromatic group up here, we can add a nitrogen, or we can add a fluorine. These types of modifications make certain groups less susceptible to being degraded by liver enzymes or other human enzymes. We can add fluorines down here, and we can replace the ester. So these were all things that we talked about and that we were hoping to do. Unfortunately, our funding ran out very quickly. I'll interject here to just to say that at the beginning of the pandemic, the Department of Energy, BER, Biological and Environmental Research, funded a bunch of national labs, I think 9 or more, to work together to work on coronavirus related research. And I was a part of that from the beginning. Unfortunately, it only lasted 9 months or so, and we were able to stretch the funding a little bit farther. But then, you know, that was kind of where some of this ended. We had to try to piece together resources and work with others outside of the lab. And we did end up getting a little bit more internal funding from ORNL through the Technology Innovation Program, which was a huge help to keep us going for a little bit longer. But unfortunately, we're still working with the group at Northeastern University to try to make molecules like this. And just in the last month or so, a preprint was published online that describes this molecule. This is from a company called Postera, and they've partnered with Pfizer. So you look at their molecule, and you can see this region here in black is very similar to the corresponding region of our molecule. You can see they've got the cyclopropyl that we suggested in our paper. They've modified the aromatic ring to have a nitrogen, and then they've added a couple of other things that also make sense. Their molecule is not covalent. You see, it doesn't have the linker and it doesn't have the electrophile, but this molecule has great potential. I expect to see a paper published in Science or Nature pretty soon. That is the culmination of this work. So, you know, it's a little disappointing, but it's also encouraging that we're on the right track, that some of the technologies or the approaches that we used were definitely going in the right direction. So I mentioned that this is in preprint stage. If you're not familiar with preprints, these days people put their paper on a preprint server before it's peer reviewed, and then they submit the paper, and eventually it will get published. So that's the stage that this one is in. I expect that it will be published, you know, in a few months or so. So just to wrap things up with this part of the talk, you can see that there is a large number of people that are involved. So Brian Sanders synthesized most of the molecules. Stephanie Delaney did a lot of the testing at ORNL. And I sort of led the project from beginning to end. Even when the funding was gone, it still had to, or most of the funding was gone, still had to get it done. This is an old slide. It was eventually published, I think in March of last year in Nature Communications, which is, you know, not a bad place for it to end up. We were happy with that. But there are people from several different national labs: Lawrence Berkeley, Brookhaven, Oregon, Stanford, SLAC, and am I forgetting anyone? Of course, ORNL, multiple universities. Well, you can see right here, Los Alamos also. Northeastern University was a really prominent one, UT, and then a company Progenera. So about 30 something authors, a lot of different resources, a lot of moving parts. And it was very satisfying to bring it to completion, even though we weren't able to deliver a drug to the market. But as you probably know, that takes generally $1 billion and 15 years sometimes. So I consider it a success story. It's something cool that you don't normally hear about from ORNL, but we did take advantage of a lot of the capabilities that we have at the lab. I'm going to stop there. It's 12:44. I'm happy to take questions. I do have some other slides on a totally different topic, but I will leave it to the organizers to tell us what we're going to do next. Yes.
The compound you mentioned, you dismissed because of toxicity.
Yes.
12 or whatever.
That's right.
Do you know the nature of the toxicity, and do you learn anything from why it's toxic? And was that done with the monkey cells?
That was done with the monkey cells, yeah. That was done at UT Health Science Center. It was really just the one result that we got. We didn't investigate the toxicity. We suspected, or we sort of guess that it's just off-target reactivity. So it has an alkyne, which maybe is a little too hot of an electrophile, a little too reactive, but we can't definitively say what else it's binding to. Yeah, good question. Other questions? Yes.
I guess sort of related to that, you indicated that Paxlovid has this liver inhibitor
Yes.
in it. Did you, the ones you found that were being broken down too quickly, could you have taken the same approach and made those improve the efficacy for those?
Certainly we could have. We didn't get to that stage. But just looking at our intrinsic clearance values from the liver microsome assays, we're not at the level that's quite low enough to start thinking about that. But definitely if we could push the clearance values lower, then that's a thought. That's something we would consider. Another thing I didn't mention is oral bioavailability. If you're familiar with remdesivir, how do you take remdesivir? Does anyone know?
Injection?
Yeah, IV. You have to be hooked up to an IV. So you can't get that at the, at Walgreens, go home and pop a pill. You have to be some sort of an inpatient or, in the hospital. And so oral bioavailability is critical for real world drugs. And Paxlovid is one of those. You take it, I think, 5 days, 2 or 3 doses per day. Does anyone remember? That's right. Something like that. It tastes terrible. That's what I've heard. So it's unpleasant. That's another reason we need to come up with a PLpro inhibitor, right? So you don't have to have this awful side effect. But anyway, so oral bioavailability, we did do some tests with our molecules and they're not orally bioavailable. They don't get absorbed and go to the right place. So there are ways that you can engineer a molecule to have the right properties to be able to do that. And we hoped that some of the modifications that I mentioned toward the end of the talk might improve that. But this molecule from Postera and Pfizer, it does have good oral bioavailability. So you can see that these types of modifications would be at least somewhat favorable. Yes.
The Postera Pfizer product, was that based directly off what you had done and they took it further?
I don't know. Their preprint is very vague. I was glad that they did actually cite our paper, but they cite it in passing. They say several other groups have developed PLpro inhibitors, but they're all poor, or something like that. Thanks, guys. But anyway, they also are very vague on their methods. They say that they used this AI approach or some sort of computational approach to develop it, but there's no description, no training data, none of the details. And I suspect that when the paper comes out, they will have to include that. But if they read our paper, they already know about the suggestion to add the cyclopropyl and to modify the ring. So who knows? They didn't say exactly what they did.
How about drug interactions with things people might be taking, like Tylenol or
Yeah,
Advil, or likely at the same time?
I mean, it's a huge concern, and we are obviously not equipped to handle all of the aspects of pharmaceuticals in our group at ORNL. We're very much on the early end, and that's more of a late stage drug discovery topic. But absolutely, it's critical, because if you have a molecule that inhibits liver enzymes, for example, then other drugs that someone's taking, their effect gets amplified, and you can effectively overdose on them. So there are things like that, and there are many other examples. That's just the first one that popped into my head. But yeah, it's a huge issue. A couple of questions over here.
I had one that, since I've had molnupiravir, I'm wondering whether, I guess it was quite a fascinating story for them to get it, but it took them a little bit longer to find a result. Did they use any of your research?
Not for molnupiravir. No, they were ahead of us. I'm going to go back to that slide because I want to say a little bit about that one. Sorry, it's a bit slow. Lots of clicking here, but there we go. Molnupiravir, that's the one you took?
Yeah.
Okay, that's from Merck, and it is still prescribed some, but it's not viewed very favorably. So I'm glad you emerged unscathed. What I know about it is that it induces catastrophic mutations to the virus.
Oh.
Now, don't quote me on that. That's what I remember reading. And so, well, what's a potential problem with that? If you don't knock it out, then it could lead to new variants. And we don't want that. They're already emerging anyway. But it does work. It does kill the virus. It just has some aspects that are maybe not ideal. So, but I'm not endorsing any particular product. It's just what I remember reading, and correct me if I'm wrong. Yeah, yes.
Yeah, one of our virtual listeners says, both times I had COVID, I became anemic and had to have iron infusions. I was not anemic before that. What might be going on with proteins to cause this?
Whew, okay, that's out of my league, to be honest with you. I don't know, but the virus just disrupts so many systems, so many different aspects of our immune system and other systems in our body. I don't know what the mechanism or cause might be for COVID-induced anemia. I really have no idea, but hopefully once you got over it, that you return to normal. Yes, in the back.
Has this work had any impact on the so-called vaccines that we've been giving?
Well, vaccines are a contentious topic, of course. I'm pretty sure that they saved quite many lives, or reduced the severity in lots of people. But people have strong opinions about them. I think people have much less strong opinions about a small molecule drug. People are much more inclined to take Advil or Paxlovid or something like that. So it's an alternative. The vaccine is generally taken as a preventive, right? Something before you're exposed to the virus to train your body to recognize it and respond rapidly. These are molecules that would be taken once someone is already infected. So it's really just a different, different beast, I guess. But my intention is not to put 5G or any sort of microchip into anybody. It's just to help them get over their COVID, you know. So lots of chuckles there. Yes.
I'd like to hear about the mercury work.
All right, okay, so it is 12:54. How much time do I have before people start getting tired and grumpy? Tired of hearing from me?
15 minutes.
15,
Yeah.
okay. So let's do that. Now I have to scroll all the way back through my slides. Any other questions while I'm scrolling?
It says compound 7 was not effective with Omicron or Delta strain. Any ideas?
Not as effective. That's actually this slide right here. Not as effective. We don't know because as far as I understand it, PLpro has essentially the same sequence, if not an identical sequence, in the different strains. So there's something else that the organism is doing, that the virus is doing to limit its efficacy, and I do not know what that is. All right. Oh, I should have mentioned that this work was funded by the Department of Energy through something called the National Virtual Biotechnology Laboratory, which was this consortium of several different national labs. Okay. Who was here 10 years or so ago when I gave a talk about bacterial mercury methylation? Anyone? Okay, good. So this is, was there one more hand? Okay, a couple of you. I'll try to make this efficient. Again, if you have questions, I can stay and talk as long as you like. So mercury is an issue here in Oak Ridge. There is mercury pollution from mostly from the 1950s and 60s. And Dave, you're welcome to correct me if I say anything that's incorrect, because you know probably way better about the history than I do. But anyway, feel free to interject. You won't offend me. So mercury was used as a solvent to separate lithium isotopes in bomb production back in the 1950s and 60s. There was a whole lot of it that was brought in, and over that period of time, some of it was lost to the environment, maybe through spills or possibly evaporation or leaks, different things like that. And so that mercury, a lot of it is still around in the groundwater, under the buildings, in basements and things like that. And a certain small amount of it, yes, do we have a?
Just a remark,
Sure.
it's generally not understood that the vacuum pumps, the huge vacuum pumps that were used for the county funds in World War II and for a couple of decades after were mercury diffusion pumps. And so the large buildings at Y12 had large storage of mercury in the buildings. And there was a famous spill, for example, in Beta 3, where mercury flooded down the stairs into the crawl space underneath and is probably still with us. But the amount of mercury involved in that pumping was vastly greater than the other lab things like the lithium.
Oh, is that right? Okay. Thank you for clarifying that. I hope that everyone was able to hear that. If not, you gotta go talk to him afterward. Okay. So everyone knows about Y12, just down the road a little bit. And some of the mercury that escapes from the Y12 plant comes out of the creek, East Fork Poplar Creek. And it's just a tiny amount each day. If it rains really hard, then a bit more comes out. But that's really where the legacy waste comes from. And so when I came to ORNL back in 2008, this program had just started to study the biogeochemistry of mercury in the environment in this area. I was a postdoc, so I had no idea about mercury. I really didn't. I mean, I knew what it was, and I had some understanding of chemistry, but that was a new topic to me. But I thoroughly enjoyed working on that for 14 years or so. Okay, so there is a global mercury cycle. Mercury gets converted or interconverted among various forms. So mercury 0 is the stuff that people put in their hand in the past. I don't know if people do that anymore. Who has had mercury in their hand before?
Yes.
Yeah, okay. And you live to tell about it. So that's important because mercury 0 is relatively inert. But when it gets oxidized to mercury 2, then it all of a sudden has a very strong affinity for various chemical groups, particularly sulfur containing groups. And so that's a problem. Because once it binds, it basically doesn't want to let go. It can, but it's very difficult to remove it. Well, that mercury 2 gets deposited into the environment somehow. And then the part that I'm going to focus on today is it can get methylated. We still don't know why bacteria methylate mercury. If anyone knows, please tell me, because I'm really curious. But certain bacteria, not all of them, certain anaerobic bacteria and archaea can put a methyl group on mercury 2. And so then you get this stuff, methyl mercury, it's got a formal plus charge. And that also likes to bind to sulfur. For example, cysteine that we talked about in the last part of the presentation. And so when it binds, it causes all sorts of biochemical problems. All right. So methyl mercury and inorganic mercury can bioaccumulate or concentrate in fish. And if people eat fish that are loaded with methyl mercury, well, they assimilate that into their bodies, and that can be toxic, particularly for people who eat a lot of sushi, for example. And I think we're all safe, but don't eat sushi every single day, particularly from large fish, because people who have done that have had real problems. Okay, well, back to the science, back to the microbiology. What I show here is a phylogenetic tree. It's basically just the interrelatedness between different species or strains of bacteria. These are all closely related, and the tree is color-coded by whether a particular species or strain makes methyl mercury or doesn't. And what you see is the blue ones, the methylators, are interspersed kind of sporadically throughout, and the black ones, we don't know what they do, or we didn't know at the time. This was 13 years ago. But there doesn't seem to be an obvious relationship between, hey, all these bacteria that are closely related, they all make methyl mercury. It's sporadic. So the question was, are there genes for mercury methylation? And of course, the answer is yes. And we discovered them. How did we do that? Well, back in the 60s, it was discovered that vitamin B12, so this is, everyone knows what vitamin B12 is. We couldn't draw the structure from memory, neither could I, but everyone's heard of it. Vitamin B12 is an essential vitamin. Somebody figured out that the methyl form of that could transfer a methyl group to mercury. And that was an important development. Fast forward to the 1990s, a group figured out that this biochemical pathway that involves vitamin B12 seems to be involved in making methyl mercury. And going back to structure, because I'm very interested in protein structures, this particular protein, it's called a coronoid protein. That just means that it binds vitamin B12. This is not involved in mercury methylation. It's involved in this other closely related pathway. Its structure was published, and so we knew what this protein looked like. Well, I got a hold of that and I said, could a similar protein that also binds B12 be involved in making methylmercury? And so that's where the bioinformatics came into play. So we had the genomes of, at the time, 6 different mercury methylating bacteria. So I showed you on the last slide this phylogenetic tree, or this family tree of bacteria. Well, we have the genomes of these 6 that all are known and confirmed in the lab to make methylmercury. And so we said, well, what is it about all of these proteins, or any one of them actually, that has something to do with this biochemical pathway? It's kind of like this protein, but it's got to be different, because we know this protein has nothing to do with methylmercury. So that was the eureka moment. I put all this stuff together and searched for a protein that had a similar sequence, similar amino acid sequence to this one, and but was present in all of these bacteria. And we found not just one gene, but 2. So the thing that's shown here in purple is just a diagram of what we eventually named HGCA. So Hg for mercury, and C for carbon, A, and then it turns out there's a B. That's another protein. It's totally different, but it always shows up right after HGCA. Sometimes there's something in between, but it's always there. And so that was pretty exciting stuff. This was in 2012. We went and told the experimental people, because I'm useless. I can't go in the lab and knock out genes or test bacteria. But my colleagues at ORNL and at the University of Missouri, they could do that. And so they knocked out this gene, what we call HGCA, and they showed that when it's present, the bacteria makes a ton of methylmercury. But when you knock out HGCA or HGCB, actually, I think, or both, you get 0. So you totally abolish mercury methylation. And then when you put those genes back into the organism, then it makes it again. So that's the evidence that we needed to show that these genes were involved. All right, so then we started making the 3-dimensional models. This is 2013, 2015 range. It's just part of the protein, HGCA, with vitamin B12 bound to it. There's a cysteine. You can tell I like cysteines. They do interesting chemistry, bound to the cobalt atom in vitamin B12. So that was an early model that we made. And our collaborators in Missouri did a lot of mutations of specific amino acids. I'm going to go back. They mutated all these different amino acids, including the cysteine, to alanine, which is sort of a bland, boring amino acid. And they found that the cysteine is really all I want to point out here. The cysteine is absolutely essential. That's doing some really interesting chemistry. And there were a few other amino acids that are very important. They're probably more structurally important, but the cysteine is chemically important. So that was a nice result. Well, then around the early COVID days, an approach was developed, not by us, but a group at the University of Washington, to predict the structure of proteins from its sequence, actually by combining hundreds or even thousands of sequences. So this is a huge phylogenetic tree of thousands of HGA sequences. And these were taken from metagenome samples. So a metagenome is when somebody goes out into the environment, scoops up sample, and sequences every organism that is in that sample. And then they throw away maybe the fungi and the plants and just keep the bacteria in this case, and then search for only the proteins that look very similar to HGCA in terms of sequence. And you can use that information to, I'm not going to go into detail, I didn't provide a slide, but you can use what's called a coevolution analysis to predict which amino acids in the protein are close to each other. And the way that you do that is when one amino acid changes in a protein, another one compensates. And so these compensatory side-by-side interactions suggest that those two amino acids are, even though they're far apart in sequence, they're actually close to each other in three-dimensional space. And so you can generate what's called a contact map, which is what I show here. And you can do that not only for a single protein, but for a complex. So this is HGCA here in blue and HGCB in purple. And you can predict the interactions between these two proteins. Now that approach was really powerful in 2017 to about 2020, but then along came something called AlphaFold. Has anyone heard of AlphaFold? Yes, one of you. Okay. Scientific discovery of the year, 1 or 2 years ago. It's from Google DeepMind. So DeepMind is a subsidiary company of Google, and they took this way farther. And now any one of you could go onto a website, put in an amino acid sequence and hit go, and out will pop a three-dimensional structure in 10 minutes or so. And it's really amazing. There's a lot of hype associated with it, and some people get tired of hearing the hype with AlphaFold, but it's really just amazing. So this is a model that was predicted pre-AlphaFold, but we went back and made a model with AlphaFold, and they're very similar. So it was encouraging that what we did was on the right track. So what do we do with the three-dimensional model of these two proteins? Well, it turns out that it's very hard to make these in the lab. How am I doing on time? A couple more minutes. Because for one thing, these two proteins are embedded in a membrane. So they're not just floating around in the cell, they're embedded in the cytoplasmic membrane. It's hard to work with membrane proteins. These also are proteins from strict anaerobic bacteria, which means that if they're exposed to oxygen, they're dead. And the same thing with the proteins. Remember, it's got the vitamin B12 cofactor, this group that binds to it. HGCB has iron sulfur clusters. So these are cube-shaped non-protein bits that bind to the protein, and they're involved in electron transfer. And producing enough of this protein to characterize it with something like X-ray crystallography has still not been done after 10 years, despite lots of effort. And so having a model helps us to infer things about the chemistry and the mechanism in the absence of having an experimental structure. And so that's why we did it. Now, there are some things that we can do in the lab to show that when we produce the protein, it does actually bind to cobalamin, and it does actually bind these iron sulfur clusters, and that was done by our collaborators at the University of Michigan. So we have some validation of our model. It's not just a prediction that's nonsense or garbage. It's validated, but we don't have an experimental structure. So that allowed us to propose a mechanism of how this methylation works. You don't have to follow all the details, but these cysteines are very important because, as I said at the beginning, they bind to mercury very strongly. But what's interesting about cysteines is if you have some cysteines over here and you have some cysteines over here, and this one is bound, these are bound to mercury, they can easily transfer the mercury to another pair of cysteines because they have similar affinity or strength of binding between the two. So you can hand it over. And so there's a pathway, we think, that mercury gets handed over to the right place so that it then sits in a position to accept this methyl group, which is shown in the black sphere. And that methyl group then gets transferred to the mercury. That makes one of the cysteines let go. So now we have this cysteine bound to methyl mercury in gray and black. And if we go down to the bottom right, that methyl mercury gets handed off from one cysteine to another. And then eventually it goes, we don't know where it goes after that. but we know how it gets into the binding site and how it gets methylated and how it gets out of the binding site. We still don't know why. Why do bacteria do that? Why do they stick a methyl group onto mercury? Does it help them get rid of the mercury? Maybe. Is it a defense mechanism? We don't really think so, because bacteria that make methylmercury are no less susceptible to mercury toxicity than bacteria that don't, which is interesting, right? Is it a weapon? Do they make methylmercury and stick it out in the environment, and that kills their competitors? Maybe. Yes.
Dynamic question.
Sure.
Mercury is such a heavy atom, and your models look at the dynamics of proteins and so on, their vibrations and what have you.
Yeah.
Could it be a dynamic effect?
So these are static models. I mean, we do have some wiggling around of the models a little bit, because when you build models with one random seed, you'll get one model, and when you build another one, you get a slightly different model. That's not really dynamics though. It's just uncertainty in the model, really. I mentioned that I've done quantum chemistry. We do use quantum mechanical approaches to calculate the reactivity of some of these models. And yes, we do have to take into account the relativistic effects, because Mercury 0 has 80 electrons, and some of those in the inner core are traveling at more than half the speed of light. So, you do have to have an appropriate level of theory to describe the reactivity. But in terms of dynamics, I have not really done much on that topic, but it's a valid point. Yeah. And so, just to wrap things up finally, that's my last slide. The mercury work it lasted over 10 years, I think from 2008 till about maybe 2019 or so. And so I'm no longer working in that area. And a lot of the reason is that the project had sort of run its course. We made one of the biggest discoveries in the field in a couple of decades or so, and new people have started working in this area and sort of picked up where we left off. But it was a fun project to work on for a lot of my career, and so I was glad to be a part of it. Even though like I said, I didn't know anything about mercury before I just jumped in and started giving it a shot. So thanks a lot for all your attention and great questions, and I'm happy to answer more.
Well, we are sort of.
Are there any practical applications like preventing methylation?
Yeah,
Is that a possibility?
that was my hope. Our task was not to.
That's what I figured.
was not to come up with
Siri.
Siri, leave me alone. No, our job was to do the fundamental research. Now there are applied programs like the environmental management program that are tasked with actually doing cleanup and remediation, things like that. That was not part of what I was doing, but practical applications. One thing that I think is really important is we now know the genes that are responsible. We can go into any environment, anywhere in the world, dig up a scoop of soil or seawater or whatever, even the human microbiome, and search for those genes to see if they are there. And so if they are, then that's a red flag. We need to be careful about mercury in this area. There are ways that mercury could be sequestered, maybe sopping it up with some sort of an absorbent material or like that. As far as actually stopping bacteria from doing it, got to be careful about unintended consequences, things like that. Good question, though. Anyone else? Had enough? All right,